At 11:07 on a Thursday night, the bottle keeps turning in his hand. Not because he has forgotten the label. Because medicine has reached the edge of what it can offer him. Across the table, his son keeps highlighting the same biology sentence, pretending not to hear the silence. And that is the anomaly. We can model proteins, generate molecules, and run AI on vast chemical libraries. Yet a drug can still look perfect in software and fail in a human body. A molecule can look perfect in software and still fail in humans. Biology is the part the model does not fully see. That failure is expensive. A single approved drug can cost roughly $879 million to more than $2.2 billion, and the full path usually takes 10 to 15 years. For families like this, that is not a budget line. It is time. Recent data from 2014 to 2023 says only 6.7% of drugs that enter phase 1 ever reach approval. That is lower than the older 9.6% figure from 2006 to 2015. So the modern system is not failing at the end. It is failing almost everywhere. The question is not whether AI can generate candidates faster. It can. The question is why speed still does not reliably become medicine. That answer starts with what a drug actually has to do inside a body. If first principles science like this is your thing, CivilizationX is built for it. Drug discovery is a hostile optimization problem. First, scientists choose a target, a protein, pathway, or cell process linked to disease. Then they search for molecules that bind it, survive digestion or circulation, reach the right tissue, avoid healthy cells, and still change disease in a real patient. Every step is noisy. Cell data is incomplete. Animal models are simplified. Human biology is diverse. And the body is not one system. It is hundreds of interacting feedback loops. That is why AI matters. It can rank targets, generate structures, and filter toxicity earlier. But here is the part that changes everything. Preclinical attrition is brutal. Only about 11% of compounds move beyond that phase, which means roughly 95% fail before they ever become a real drug. And once candidates reach human testing, the biggest reason they die is not toxicity. It is efficacy. Recent analyses put lack of efficacy at 56% of phase 2 and 3 failures, with safety at 28%. That means the molecule may be chemically elegant and still do nothing useful in people. AI helps by compressing the search. It learns patterns from chemistry, protein structure, genomics, and toxicity data. It can predict hepatotoxicity, cardiotoxicity, and genotoxicity before expensive wet lab work. It can propose new scaffolds. And some AI-designed candidates have reached human trials in under 18 months instead of 4 to 5 years. AI-native firms are even reporting phase 1 success rates around 80% to 90% for these candidates. But phase 1 is only early safety. It does not prove the drug works, and no novel AI-discovered drug has full approval yet. Scientists soon discovered something unexpected. Structure prediction was a huge win. But structure is not destiny. AlphaFold could predict protein shapes with unprecedented accuracy, and that transformed target work. But a static fold is not the same as a living pathway, a dosing schedule, or an immune system responding in real time. The chemical universe is bigger than 10 to the power of 60 possible molecules. Existing AI has explored less than 10 to the power of negative 34% of it. So the problem is not lack of ideas. It is deciding which ideas deserve the body's limited testing time. But solving that created an entirely different problem. AI has pushed the bottleneck forward. We can now generate target and molecule hypotheses faster than we can gather high-quality integrated human data to prove they matter. That is the translational gap. But here is the problem. A model can be excellent at pattern recognition inside its training data and still miss rare biology, off-target effects, or patients whose physiology does not look like the average case. That gap hits hardest where data is weakest. Rare disease patients, underrepresented ancestries, unusual comorbidities, and fragmented medical records are least visible to the models. So, the people with the fewest treatment options are often the ones the system understands least well. That is not a software bug. It is a dataset problem with human consequences. So what has AI actually done? It has made early discovery faster, cheaper, and more focused. It can remove weak compounds sooner, reduce wasted synthesis, and prioritize targets with better evidence. As Charles Lin at Amgen put it, AI helps scientists work faster, but real progress still depends on human judgment. That is the right frame. Not AI replacing biology. AI helping us ask better questions of biology before patients run out of time. The next 5 to 15 years will not be about fully automated drug invention. It will be about better translational prediction. Models that combine multi-omics, chemistry, toxicity, and patient stratification well enough to cut late-stage failure. The near-term milestone is not magic. It's a carefully measured, tangible step. It is better feedback loops between models, robotic experiments, and human data, so fewer false leads survive. Back at the kitchen table, the bottle still turns in his hand. What failed this family was not effort or intelligence. It was a system that could generate thousands of promising ideas without reliably knowing which one would work in a body like his. That is the real question now. But here's the counterintuitive part. What kind of experimentally grounded model would be needed to predict, before phase 2 begins, whether a drug candidate will be effective and safe across biologically diverse patients rather than only in simplified preclinical systems? What do you think would have to change first? The data, the models, or the experiments? The constraint is real. The data exists. Whether the systems built around it can act on it at scale, that is the next engineering problem. Stay with CivilizationX.